Hi, my name is Kier. For today's video we will be talking about the principles of fermentation. First off, we need to define first what is fermentation. Fermentation is the chemical transformation of organic substances into simpler compounds by the action of enzymes, complex organic catalysts, which are produced by microorganisms such as molds, yeasts, or bacteria. The enzymes from the microorganism act by hydrolysis, a process of breaking down or pre-digesting complex organic molecules to form smaller, and in the case of foods, more easily digestible compounds and nutrients. Now that we have identified and understand the basic mechanism of fermentation, I think it's about time to help you recognize the different types of fermentation process. There are two types of fermentation process which are based on respiration: aerobic fermentation and anaerobic fermentation. Both types of fermentation require two primary components, a sugar supply and a bacterial culture. Let's discuss one by one and identify the differences between the two processes. Aerobic fermentation occurs in the presence of oxygen. It usually occurs at the beginning of the fermentation process. Aerobic fermentation is usually a shorter and more intense process than anaerobic fermentation. Wine, beer and acetic acid vinegar, such as apple cider vinegar, need oxygen in the primary or first stage of fermentation. When creating acetic vinegar, for examples, exposing the surface of the vinegar to as much oxygen as possible, creates a healthy, flavorful vinegar with the correct pH. Anaerobic fermentation. Anaerobic fermentation, which is common to all bacteria and eukaryotes, is a metabolic process that converts carbohydrates, sugar, to organic acids, gases or alcohols under anaerobic conditions. Anaerobic fermentation has a broad range of applications. Two common examples are ethanol fermentation and lactic acid fermentation. Lactic acid fermentation. Lactic acid fermentation is a metabolic process by which glucose or other 6-carbon sugars, also disaccharides of 6-carbon sugars, for example sucrose or lactose, are converted into cellular energy and the metabolite lactate, which is lactic acid in solution. Example of lactic acid fermentation are kimchi and pickled vegetable fermentation. Factors affecting fermentation. Temperature, pH, and substrate concentration, all influence the fermentation process and metabolic processes. Temperature. Temperature is an important factor for microbial growth. All microbes have a certain optimum range in which they can grow. As the temperature rose, the maximum fermentation time decreased, but a much higher temperature inhibited cell growth and then significantly reduced fermentation because the higher temperature changes the transport activity or saturation level of soluble compounds and solvents in the cells, potentially increasing the accumulation of toxins such as ethanol inside cells. pH. pH is a component that could affect cell development as well as alcohol yield. It is critical to maintain a pH range of 4.0 to 5.0, but if it exceeds this range, the production of byproducts such as acetic and butyric acid may have eaten part of the substrates, lowering fermentation efficiency. Substrates type and concentration. Different fermentation patterns occur depending on the substrates. Increasing substrate concentration also increases the rate of reaction to a certain point. Once all of the enzymes have bound, any substrate increase will have no effect on the rate of reaction, as the available enzymes will be saturated and working at their maximum rate. And now we reach the end of the slide. I hope you learned something. Thank you for listening.